This week we will be talking about water quality testing. As most of you probably know, water is an essential resource. We absolutely need water to survive. And you may have heard of the fact that there's a water shortage. That doesn't necessarily mean that there's a shortage of water per se. The problem is with the shortage of clean water and water that is accessible to those who need it. So unfortunately, not all water looks like what we have on the top right, where we have this, beautiful waterfall and all of that. What you are probably, unfortunately, more familiar with, is looking at contaminated rivers. Probably not fish dying off to the point what you see on the top left, but um, there's lots of different ways in which we contaminate water. We do it through agriculture. We do it through our waste. We do it through industries. And there's different types of pollution that you can create, all of which is harmful to the creatures who call the water home, and eventually to us. The quality of water is affected by a number of different things. Those things are things like the temperature, the pH, the conductivity of the water, the amount of dissolved oxygen, its hardness, nitrates, phosphates. And so this is more of an effect rather than a cause. The quality of the water affects the biological profile. And then, of course, the existence of bacteria in the water can be a sign of low quality. So variations in temperature. Different organisms have different ranges of temperature that they will tolerate. And so sometimes it's a problem when humans create thermal pollution. So that's actually a pollution. Those could be that the water is just simply too hot for anybody. It could also be that certain enzyme functions work better at some temperatures than others, and those are sometimes problematic. There is thermal pollution, thermal variation in water that is normal. So in the summer, the surface water tends to get hot. And so heat intolerant organisms move deeper. And in the winter, the surface water gets cold. So cold intolerant organisms move deeper. And you've probably all experienced this before when you jump into a lake or swim in a lake or whatever, where there's not much movement of the water, so there's not much exchange of the water. There are very clear layers of temperature that you can experience, and a lot of times, even though the surface water is pretty warm, um, where your feet are, when you stick your feet down, it gets quite cold, and that's very similar to the way dirt is too. That the further down you get, the more constant the temperature is. Now, this temperature also affects water chemistry, and I'll talk in a little more detail about that in just a minute. So one of the things that's affected by temperature is the pH of the water, and that measures the acidity or alkalinity of the water. When we measure pH, it's a logarithmic scale from 1 to 14. Logarithmic means that a pH of 2 is not double the pH of 1, it's 10 times the as basic as a pH of 1. It's important for the activity of enzymes and other physiological functions of organisms. And to just give you an idea of what kind of range we're looking at in terms of pH, you have it starts with a pH of 1 is something like stomach acid or your lemon juice or your vinegar, those kinds of things. whereas pure water is a pH of 7. I want you to remember that. And then hydrogen peroxide, those kinds of things, those are the higher pHs. Conductivity is the capacity to convey an electric current. So it's usually related to the concentration of ionized materials in the water. The standard unit of conductance is MHO, which is ohm spelled backwards, since conductance is the reciprocal of resistance, and scientists have always had a sense of humor. It's reported as it's measured as Siemens. It's reported coming from the name Siemens. Reported as MHOs per centimeters or Siemens per centimeters. Conductance is one of those things that varies with temperatures, just like pH does. So you have to measure conductivity at a given temperature. So usually you say it's got to be conductance at 25 degrees Celsius. In distilled water, the conductance is usually between 0.5 to 2 Siemens per centimeter. Hard water, like where you have like mineral water, ranges from 100 to 1,000 Siemens per centimeters. The sewage doesn't usually change. Just because it's sewage water doesn't usually change the conductance value. It reflects the area of water supply. However, industrial wastes sometimes may exceed these numbers by a large amount. Dissolved oxygen is vital to the life cycle common to water because it's essential for fish to live in the water. It is soluble in the water. And one of the surprising things is that the solubility of oxygen goes down as the temperature goes up. So cold water has a higher distilled oxygen content, can hold a higher distilled oxygen content than warm water. Warm water fish, they can handle distilled oxygen greater 4 milligrams per liter. When the distilled oxygen, not distilled, dissolved oxygen drops below 4 milligrams per liter, you end up having fish die off. Cold water fish need dissolved oxygen of greater than 5 milligrams per liter. And a fish like trout or salmon that you tend to find in water that's cold, and also water that's like there's lots of waterfalls, which means that there's a lot of like rapids and stuff like that, which means there's lots of dissolved oxygen in there. They require amounts greater than 7 milligrams per liter of water. And somewhere in Nicholasville, I think there's they stock ponds in the winter with like trout, and the trout tends to be it's fine there as long as the weather's cold enough. But as soon as the weather warms up, the lack of oxygen in there will kill the fish unless the fishermen get them out of the pond before that. Hardness is the total concentration of calcium and magnesium ions expressed as calcium carbonate. They range from 0 to hundreds of milligrams per liter, also called parts per million, ppm. Drinking water quality standards include soft water, which is from 0 to 60 ppm, medium hard, 60 to 120 ppm. Hard water is 120 to 180 ppm, and then we have very hard water that's greater than 180 ppm. We here live in an area that has hard water, and as a reminder, that one of the reasons why I know this is because we have limestones here, and that tends to dissolve in the minerals from limestone tend to dissolve in the water. And so this is why you have this is what causes the caves to show up, right? So that's the water drips down the, which ones are the hanging ones? Anybody remember? Those are the stalactites. The stalactites hang down from the ceiling, and the water goes down those little droplets. The water evaporates, and the minerals remain behind and cause cause these formations. Nitrates are essential for plant growth, but if they're excessive, then you get pollution. And this type of pollution usually comes from sewage or agricultural fertilizers. And the problem is that it stimulates the growth of algae. It's also harmful to fetuses and young children. When you get to amounts of nitrates greater than 2000 ppm, you end up with miscarriages and deaths of newborn. And so we regulate this one relatively tightly. So the maximum contaminant level in drinking water is 10 parts per million of nitrogen. Looking at this tells you a little bit how the effects of agriculture on groundwater contamination. So if you look over there, basically in the areas where you have like the highest concentrated, like industrial type agriculture, where you have high amounts of fertilizer being spread onto large fields, monocultures, that kind of thing. You have the highest input of nitrogen, and then therefore the highest vulnerability of the aquifer. Phosphorus, very similar to nitrogen, is an important nutrient for aquatic plants. But again, excess phosphorus can lead to problems. And one of the problems that we're talking about here is called eutrophication. So we're trying for phosphorus shouldn't be more than 0.1 parts per million unless it's polluted in wastewater or in agricultural areas. So this is what we're talking about when we're talking about eutrophication. So you've got a normal lake here, and then you've got this thing that's called an algal bloom. Now, when you first look at this and you're looking at this algal bloom, you're like, it's not a problem. It's green, right? Green's good. Well, what happens during eutrophication is you have the phosphorus runoff, and it fertilizes small floating aquatic plants. The sunlight increases this, and so these small plants are very happy, right? However, the problem is the light that penetrates down is reduced, and so there's two things happen. One, the organisms further down, like the plants further down, they can't get the sunlight. Um, and then at the same time, when these surface plants finally die off, because eventually they do, because they form layers and layers over one another, and when they die off and decompose in the water, they deplete the oxygen in the water, and then the animals die because of the lack of the oxygen. So that's the problem with eutrophication. One of the ways how we can figure out if a aquifer, or if a stream or pond is healthy is we look at the biological profile. What we do there is we collect animals that are living at that locality, and we check how many are there and under what conditions they exist there. And some indicators, some species are indicators of pollution that is present. For example, midge larvae have hemoglobin in their bodies that helps them transport oxygen. So they can live in water that's quite low in oxygen. So if you find a situation where you don't have a lot of animals, but you've got a whole bunch of midge larvae, that's probably water that's very low in oxygen. So what you're looking at when you're looking at a biological profile, you're looking at the correlation between the number of species present and how much you have of each of those species. In a high quality environment, you have it's usually a stable environment where you have many different species, but not very many individuals per species. Whereas in low quality polluted environments, you have fewer species, but high numbers of individuals per species. And I'm a big fan of wildflowers. And so when you're looking at this field down there, which one is probably the most high quality environment? It's not the one on the right, right? Now, when you're looking at bacteria, the one that you're looking for most of the time is you're looking for E. coli, Escherichia coli. Because what you're mostly interested in is you want to know if there's fecal contamination. And so E. coli, Escherichia coli, is a fecal coliform bacterium, coliform because it comes from the colon. They are the indicator organisms for that. So they're not themselves the pathogens, even though some of them can cause diarrhea. But what they do is they show you that there has probably been fecal contamination. Um, and with fecal contamination come other issues because you know that there's been human waste that has contaminated the water, and then human waste comes with its own problems. So the way you test for these bacteria, you add a mill of water to test media, you incubate it at room temperature overnight, um, and if there's bacteria present, the broth in the media will go from dark green to yellow. However, that's only a presumptive test. You'll need further tests to identify the actual organism. And that's it for the lecture. And you can go continue to the lab materials.